Welcome to today's lecture on the topic of antimicrobial peptides, AMPs, as novel therapeutics. In this detailed script, we will explore various aspects related to AMPs, including their classification, mechanisms of action, structure-activity relationships, design approaches, resistance, and their application in the treatment of bacterial infections, antifungal and antiviral activity, immunomodulatory actions, peptide delivery systems and formulations, pharmacokinetics, as well as clinical development and translation. So let's begin. Antimicrobial peptides, AMPs, are small molecules found in organisms that defend against microbial pathogens. They have broad-spectrum antimicrobial activity and can target bacteria, fungi, and viruses. AMPs have unique characteristics that contribute to their antimicrobial properties and are being explored as potential alternatives to conventional antibiotics. They can effectively combat drug-resistant pathogens and modulate the immune response. AMPs can be classified into different families based on their amino acid composition, structural features, and mode of action. Some common classes of AMPs include defensins, cathelicidins, and histatins. Mechanistically, AMPs exert their antimicrobial effects through multiple targets and mechanisms, including disruption of microbial cell membranes, inhibition of cell wall synthesis, interference with intracellular processes, modulation of host immune responses, and induction of apoptosis. The diversity in their mechanisms enhances their ability to combat various microbial pathogens effectively. The structure of AMPs plays a crucial role in their antimicrobial activity, and understanding the structure-activity relationships is essential for designing potent peptides. AMPs often adopt secondary structures like alpha helices, beta sheets, or extended conformations, depending on the environmental conditions and the target pathogen. The degree of cationicity, amphipathicity, and hydrophobicity, as well as specific amino acid residues and sequence motifs, contribute to their efficacy. Moreover, post-translational modifications and other factors influence the stability, selectivity, and toxicity profiles of AMPs. To overcome limitations such as susceptibility to proteases and low stability, several strategies have been employed to optimize AMPs. Rational design approaches involve the modification of peptide sequence, incorporation of non-natural amino acids, cyclization or stapling, conjugation with targeting moieties, and hybridization with other therapeutic agents. Furthermore, the use of computational tools, such as molecular modeling and molecular dynamics simulations, aids in predicting the structural features and optimizing the properties of designed peptides. While AMPs are generally effective against a broad range of pathogens, the emergence of resistance mechanisms poses a challenge. Microorganisms can develop various strategies to evade the antimicrobial effects of AMPs, including modification of the cell surface charge, secretion of proteases or efflux pumps, alteration of membrane lipid composition, or production of specific resistance proteins. Understanding the mechanisms of resistance and exploring strategies to overcome or prevent resistance is crucial for the practical application of AMPs as therapeutics. The escalating antibiotic resistance crisis has fueled interest in exploring AMPs as alternatives for treating bacterial infections. AMPs have shown promising efficacy against drug-resistant bacteria, including methicillin-resistant Staphylococcus aureus, MRSA, vancomycin-resistant Enterococcus, VRE, and multi-drug-resistant gram-negative pathogens. Their ability to target and disrupt bacterial membranes, interfere with essential cellular processes, and modulate the host immune response makes them attractive candidates for combating bacterial infections. In addition to their antibacterial properties, AMPs also exhibit antifungal activity. These peptides display effectiveness against a wide range of fungal pathogens, including Candida albicans, Aspergillus species, and dermatophytes. The mechanisms of antifungal action vary among different AMPs but commonly involve targeting fungal cell membranes, inhibiting cell wall synthesis, disrupting intracellular processes, and interfering with fungal biofilm formation. AMPs offer potential alternatives for the treatment of invasive fungal infections that are notorious for their resistance to current antifungal drugs. AMPs have demonstrated antiviral properties, showing efficacy against various viruses, including enveloped viruses like HIV, influenza, and herpes simplex virus. The mechanisms by which AMPs inhibit viral infections involve direct interaction with viral envelopes, inhibition of viral entry and fusion, prevention of viral replication, and modulation of host innate immune responses. Further research is ongoing to harness the potential of AMPs against emerging viral threats and to understand their role in combating viral infections. Beyond their direct antimicrobial effects, AMPs exhibit immunomodulatory actions that are important for regulating the host immune response during infection. These peptides can activate both innate and adaptive immune responses through interactions with immune cells, such as neutrophils, macrophages, dendritic cells, and lymphocytes. They can enhance chemotaxis, phagocytosis, cytokine production, and promote wound healing, while also attenuating excessive inflammation. The interplay between antimicrobial and immunomodulatory functions of AMPs highlights their potential therapeutic applications in various pathological conditions. The development of effective delivery systems and formulations is crucial for optimizing the therapeutic potential of AMPs. Several approaches have been explored, including liposomal and nanoparticle-based formulations, encapsulation within hydrogels or micelles, conjugation with cell-penetrating peptides or targeting ligands, and incorporation into biomaterial scaffolds. These delivery systems aim to improve peptide stability, enhance bioavailability, prolong circulation time, and target specific sites of infection. Overcoming challenges such as poor pharmacokinetics, off-target effects, and toxicity profiles is essential for successfully translating AMPs into clinical practice. In order to effectively use antimicrobial peptides, AMPs, as novel therapeutics, understanding their pharmacokinetics is crucial. Factors like absorption, distribution, metabolism, and excretion impact the bioavailability and effectiveness of AMPs. Modifications, formulations, and delivery systems can also affect these aspects. Plasma protein binding, tissue distribution, cellular uptake, and elimination pathways further influence the pharmacokinetic properties of AMPs. A thorough comprehension of these aspects is essential for optimizing A.M.P.'s pharmacokinetic profiles and ensuring their clinical efficacy. The development and translation of antimicrobial peptides, AMPs, as therapeutics face numerous challenges. Comprehensive preclinical testing, regulatory requirements, manufacturing, and quality control are crucial. Clinical trials are necessary to evaluate safety, efficacy, dosing, and adverse effects. AMPs offer promise in combating drug-resistant pathogens. In conclusion, antimicrobial peptides, AMPs, are a promising group of new drugs that can fight a wide range of infections. They work in unique ways and can help overcome antibiotic resistance. However, more research is needed to optimize their properties, ensure their safety, and bring them into clinical use. Studying AMPs offers hope for finding effective and sustainable ways to treat infections.